Good evening, everyone. So we are almost second last day of our workshop today. I still have something to show you. Today's agendas will be one of the biochemistry technique that I left is crystallography, which is quite important, which is quite easy to understand though, but still I felt like before I leave you, extra crystallography would be nice and some applications then we will do from the industrial point of view, agriculture point of view, food, dairy, animals, how our recombinant DNA technology could be used in that part. So today, all in all, and also if time is left, we also go with the good lab practices, GMP, good GLP, good lab practices. So we will also deal with them and touch those part. Bioinformatics will be done tomorrow. So I had a talk with my colleague, and she agreed to do it tomorrow. If for some reason is not possible, then I will be taking that class. So don't worry. So I will teach you if I will be teaching you. So I will be dealing with FASTA, BLAST, some introduction about informatics, and if she's teaching you, then she will deal with the sequencing part also, and some more techniques that she has learned in recent years. So let's see. It's dicey, 50-50. So either she or either I will be taking tomorrow class. If I'm not taking class tomorrow, then today will be my last day of yours. And guys, listen. Uh, Share with your friends also. I will be not entertaining any files on the telegram that you're sending. Please send me via email. Please send me everything via email. uh, because it's very hard, because it's in the phone and it's quite difficult to download over the laptop, and we have a full proof, it's always good to share things over the email so that you have a proof that you have shared something with me and people will know then. So this is what I have created now. So the students who are sending me, so I have created one category for yours for reviews at Edufabrica. So I will go through them over the weekends, go through each of them and let you know what my views, how they are, and send you each one of you of your assignments and reviews, how's things are working out. So, but yeah, somebody has NEET exam, somebody got COVID positive, somebody is has its own problem. So I excuse those students, but to the most, uh, I will share it here also. So I mean, okay, I extended to 5 days, but the more you get late, the more the certification given to will be late. So try to send me ASAP after tomorrow. So today's agenda would be extracrystallography, applications of recombinant technology in various fields, and good lab practices. So these three different topics I will be dealing today. Okay. Any suggestions? If today will be my last class, I would be happy to know your suggestions just now that things need to be improved. And also, try to give your reviews on the workshops. So we will share with you links. So each workshop that you have been through, just so we will share the link with you soon. Uh, So I had this biochemistry workshop from 29 31st July. So okay, it's not working, but there were some reviews over there. So we need your reviews. So we will put for this workshop also this thing. So out of 5 stars, whatever you feel like, just give your reviews. So what do you think? Out of 5 stars, how many stars would you like to give me? In Zoom? Yeah. So we are having talk with Zoom people, but somehow we were not successful in making it happen for the 500 students. So we kept it on the YouTube platform. But from next time, I'm trying to make it on Zoom. So I will also convince Zoom people to give us make it things done good for us. 5 star. Thank you so much. Thank you so much. Thank you so much. I'm I am blushing now. Uh, but we all knows what are our weaknesses, what are our good points, where we are standing. Um, so we are trying to improve those points in our life because, and as a human being, we are always a learner, and it's never ending process. Um, and we trying to be best of our level. So try to bring this skill also in yourself. Never be satisfied with your one point. Always try to what do you say, get the best out of yourself, make the best out of yourself, and bring it to the public, whether it's presentation or sending reviews. I found really excellent reviews, I have to admit. I was surprised. Within few days, students can write such a great reviews. So you are great future scientist. You are great future of India. I'm very glad. So just go get the position wherever you looking for, and don't stop. Like Steve Jobs says, be hungry, be foolish. Just keep like that and keep going for the stars, reaching the highest stars. So, on that note, it's already 8 o'clock. Uh, Bache, there is no page limit. You can write as much as you like. But it should have all the content like introduction, um, all the basic part, discussion, conclusion, future aspects, abstract, everything should be there. No, no, Edufabrica did really nice. They handle you all students very nicely. Uh, they did. You don't know what they did, so they are under the mirror, but they really worked hard for this. Vatsal sir, Shivani ma'am, I want to give them hats off to them, 5 stars to them actually for their great efforts. And even I call them late night if I'm going through some trouble for your teaching, they're always there for me. So it's a team effort actually. It's not about me being a training. It's that Fabrica in total is working out. Yes, there is. That's why I always say, and we also know our weaknesses. I have to admit, and we try to bring that those things should not happen again and again, and trying to overcome them and get the best out of it. So, okay, lots of motivational talk we had. Let's zoom into the work part now. Where are we? We are in Edufabrica, MBBT, workshop, night day. Okay, I will. So, guys, I have to teach you today bioinformatics actually, but I had one a teacher, one tutor, one trainer from Denmark, and she had done her bioinformatics in her master's in bioinformatics from Lund University, Sweden. So I thought maybe she will be better person to teach you, not me. So I just give her an opportunity to introduce with you all students, and you can also have better overview of that subject. So today's class is postponed to tomorrow, and tomorrow class I am taking today. All right. Um, is it mandatory to write review of everyday class? It's not mandatory, but it's good for you, Paul Lamy, because you can have a very nice overview from these workshops. Because at the end of the day, you will not open these videos to check what I have learned, what I have not learned. So it will be good for yourself to have those papers with you always in a small file, just 20 page. And whenever you have examination or any interview regarding molecular biology techniques, you can go through them, and you have written them by yourself, so you will remember them in a good way. So, so something about X-ray crystallography. It's basically in this we use to identify structure of protein with the help of diffraction technique by using a molecule as a crystal, making a molecule as a crystal, and then we put X-ray light on top, and then it's got diffracted, and then according to that we predict that this might be the structure looks like. But just that's the basic principle behind the extracrystalline diffraction, and there's a Bragg's law of diffraction is being used in this technique, with the sine theta and cos theta angle are getting diffracted. So we will deal a little bit about that also, and yeah. So let's continue. So what you will learn basically the basic principle of X-rays in this topic, and phenomena of diffraction, how it works, and how it can help you to identify the structure of protein. So let you get to the in-depth knowledge of instrumentation, mechanistic overview of X-ray and crystallography. So these things we will be covering today. So why structure determination? What does it really help us to understand? So this will help us to understand how proteins interact with other molecules. If we know the protein structure, and we will get to know about catalysis is happening in the case of enzymes, interaction of proteins with other molecules, including protein itself, uh, folding, misfolding, miscoding of proteins associated with the disease. So these all parts will be dealing with the structural determination. Um, and then after the getting to know about structure, so why crystallography? Because it's a high form of resolution microscopy. It's unable to visualize protein structure at the atomic level and understand the protein function afterwards. It's basically the main principle behind is that the X-rays are diffracted by crystals. So you have your crystal here, then you give your X-rays, and uh, yeah, and then from this crystal, like here from the direct direction, it's diffracted around 20 degrees Celsius like this. So we know if it got diffracted here, so we put it the line here. So like this, all diffraction lights are being detected at the detector. So based on that, we can predict the protein structure. It might be protein could be like this. let's say if I have what do you say? This is a car, and you want to predict the structure of the car. So I will put X-ray on top, and it will diffract into this direction. So I will put the X-ray here and it will diffract into that direction. So I will observe them each diffraction onto my detector. So I will know that there is something is there. That's why it's got diffracted by this angle. That's why it's got diffracted like this angle. So just that's the main principle behind this. So by doing so, I will do it from all the directions, and I can predict. Okay, this is the backbone of the. This is the back side of it. This is the front side of the car. These are the tires. Yeah. So like this way, you can say in the protein structure that if we diffract them with uh, here, this is alpha helix structure, or if there is a beta sheet of a protein, you can predict that that might be the beta sheet, and by this you can predict the whole protein. So that's how this technique works like. So microscopy. Um, So there are three different microscopy that we have discussed so far. We have done with the light and electron. So third one is X-ray microscopy. So each has their wavelength around 310 nanometer and 1 angstrom. So it has the even higher one range, even lesser than that one, better wavelength. So better visualization. So less the wavelength, better the resolution. So in the first case with the light, we can see with individual cells, subcellular cells, organelles. But with the electron microscope, you can see cellular architecture shape of large protein molecules, atomic details of proteins. So you can go into the more atomic details with the X-rays of the protein. So why use X-rays and crystals? X-rays in the order of atom diameter and bond length, so allowing these to individually resolve. So that's how X-ray. That's why the best rays that will be. We didn't use gamma rays or alpha rays, beta rays. We use X-rays because they are in the atom shape and it allows individual things to resolve easily. And no lenses available to focus X-rays. Crystals act as a magnifier of scattering X-rays. So we don't need any magnifier in this case. Itself crystal act as a magnifier to scatter these X-rays for further results. So how it works? So first we do the protein purification. Then from the purified protein we make a crystal. Then from the crystal we collect the data with the help of X-ray crystallography. Then structural solution, phasing phase is there. Then structure determination, modeling, building, and refinement. So like this is a protein crystal. Then X-ray crystal, X-rays are been there. Then this is diffraction pattern initially that you see. Then you make phases out of this. This is a refinement place where you use your detector and computers. So you create an electron density map first of all. What you create? An electron density maps, which then fit it to the atomic model of your uh, protein. From this model, you make a final atomic model. So that's how you predict your structure. That's how what you saw in the pi mole. It's done by the crystallography, X-ray crystallography, and it's one of the hardest technique actually. I have to tell you, to get the crystal initially, a fine crystal is one of the difficult part. For some students, it's take whole PhD to create a crystal, get a refinement, and get the extra crystallography out of it. It took them whole PhD to make the steps done, 3 years or more than 3 years to do all these steps. So this is very very challenging field, but it also rewarding. Then you get If you found one protein structure which is unknown to the world, that could be a Nobel Prize winner also, because finding something new which is quite noble and good for the society could be really very, very significant for the society. So, in the first step, as we know, with the help of gels, electrogel electrophoresis, you can extract the protein. So, in the protein purification process intended to isolate one or few proteins from a complex mixture or cells, tissue, or whole organism. So, why protein purification? It helps to characterize the function of a protein, interaction of the proteins, uh, and requirements are minimum 5 to 10 milligram per soluble. So we need quite good amount of protein in this case, and proteins are required to be better than 95% purity. So more than 95% your protein needs to be pure. So next, where we were, sorry, I got lost somewhere. Yeah, the after your in the first step we purify the protein. The next step would be to to make a protein crystallized. After getting a protein, because we cut it from this gel, this protein, then we make it in the liquid form, and then getting that protein, you have to make a crystal. So crystallization will happen. Uh, so we will not go to that part because we are just discussing about more about crystallization, extra crystallography, how it works, the basic principle. So, but why crystallization is done? Because you can see extra crystallography can make a single unit to be unimaginable weak. Because single unit cell, if this is molecule, it will be hard to identify a structure with just small single structure. But if you have a crystal which is a huge number of molecules of the same orientation, so it will have like in this box 1 2 3 4, 1 2 3 4 3, the 12 on this side. So 12 on this side. So there will be a lot of chances to identify the protein in a better way actually, if you have like this more. And scattered wave add up in phase and increase signals to level which can be measured. This is often a rate limiting step in the straightforward structure determination, especially for the membrane proteins, because membrane proteins are the biggest one. So if you make this step to make a crystal, it will helps you a lot to have a better resolution. So next part, in continuation, so your crystal should be less than 1 millimeter. No crack should be there, no incursions such as air bubbles, nothing should be there. So first your crystals are grown at 4 degrees Celsius with 30% MPH, pH 6. Then crystals grown at 17 degrees Celsius, 30% MPH, pH 6. Then 17 degrees Celsius, 30% MPH, pH 5.6. Then salting setting drop at 17 degrees Celsius, 31 mph and pH 5.6. So this is how from the circle droplet it started to form into crystals like this, by decreasing the pH, by keeping the temperature at 70 degrees Celsius, and by changing a bit of concentration of our solution. So in this forming the protein crystal structure, we basically use hanging drop method, in which we use 1 to 5 microliter of protein solution suspended over a 1 milliliter reservoir containing precipitant of solution. Example, ammonium sulfate solution or polyethylene glycol. So like this. So this is hanging drop with protein. Yeah, this is hanging drop with protein and reservoir with precipitant. This is your reservoir. So here your crystal structure is being made. Then further uh, X-ray crystallography. This is the Bragg's law, I guess. This is how the first case, if your protein structure is there, you have a primary beam. It's got diffracted like that. Then you have this one unit cell. Then within this one unit cell, this is your primary being is being there. Then you have this B, C, and C will be equal to D, and that will be equals to the D dot sine theta. That theta would be uh, the angle of diffraction, and D would be the length, the width of your between two each other beams. So that's how you can calculate, uh, yeah, the distance of this Bragg's law. So I will also show you one video about this, how it works out. And then after the beam is done, everything is done. So it's time to mount your crystal. So crystals are mounted on a way that samples can be rotated and X-ray beam can be passed through the sample. So in this case, they are mounting the uh, crystal here on this top, uh, and methods of mounting included using either capillary or a tube. Both capillary tubes are mounted on a goniometer. This is your goniometer is there. So once your crystal is mounted, then it's time to expose them with the X-ray beam. You can use a synchrotron, which gives higher resolution luminosity. It's better than X-ray rays, or you can use X-ray generators for smaller laboratory use. So both are possible. Then in the data collection, source of X-rays mostly is done by synchrotron, and typical size of crystal for data collection should be of this size, 0.3 by 0.3 by 0.1 millimeter. The crystals are bounded with X-rays which are scattered from the planes of the crystal lattice, and the scattered X-rays are captured as a diffraction patterns on detectors such as film or an electronic device. So here you can see uh, a rotating anode of copper on which we put 50 kilo ke volt of electrons. Then we put them on the mirror. Then this is a primary X-ray beam. Then we focus them, these beams. Then we put them into this chi circle where the crystal is stacking there, and then there's an area detector there. So this is your circle goniometer. Kappa geometry is there actually. So afterwards, rotate crystal through 1 degree and record your XRD pattern. So you rotate your crystals for 1 degree. Then if XRD pattern is very crowded, reduce the degree of rotation. Repeat until 30 degrees were obtained. Until you rotate it to 30 degree, keep obtaining the data. Sometimes 180 degree is also needed to achieve your crystal symmetry. So lower the symmetry, more data are required. So higher the resolution, use synchrotron. If you need higher resolution, always use synchrotron. So step 4. Um, so in this, it's a structure solution that the facing state that we discussed in previous. So we have our diffraction data. Now we want to convert that into the electron density data. So this is your diffracted data. Typical diffraction pattern looks like this of a protein structure. You can see this is the protein structure, and we want to convert this into the 3D structure obtained above into electronic density map of the crystal like this. So this is all happen after the diffraction from the protein. So there some electron density was found out after the scattering, and they say maybe there's a protein structure is present. So that's why its structure goes like this, like that way. So I hope you have understood how from uh, this one, from crystal structure to protein structure, how we manage to have depicting the protein structure from the crystals of proteins. So you have learned the basic operating principle of X-rays and its application in proteomics. You are able to have understand the principle of X-ray crystallography, and you get to know about theoretical overview how to elucidate structure protein based on X-ray diffraction data. So these are some reviews if you wanna go for some references. So thank you very much. So now you have once understood this part. Now what's the time for this? A video, so that it could be better clear to you. It's video time. So you will understand each thing what you have done now in more details. So I will turn on as the go. Students, this is just a small video about Bragg's law and X-ray diffraction that we have seen. So I will continue this one. That's it. So, class, that was something basic about Brake's law and crystallography, how it works. Um, so, let's continue to the next topic. So, we are done with this part also today. Um, the applications of recombinant DNA technology in health, agriculture, environment, industry. So, this topic you might uh, we might have done that quite before also, but I want to end this presentations by bringing you to have a little bit of what we have done, how they are helping us in various spheres of field. So that's the reason I choose this presentation for today. So, how application of recombinant DNA technology in health, agriculture, environment, and industry they could be helpful to us. So, application of recombinant technology in health perspective. So, it could have a wide RNA technology have wide spectrum in improving health and also treating defected genes or introduce new ones. Application laboratory test, parental diagnosis of genetic diseases. So all these things are possible. Acquired immunity is also possible. It's also help us to acquire protective antigen proteins such as hepatitis B, influenza, HIV, mouth and foot disease. So how you can produce these vaccines, this like say hepatitis B vaccine, HB vaccine with recombinant technology. So you have your hepatitis B virus. From that you get your DNA part or RNA part. Then HB, so you have your HB antigen producing that gene. Yeah. And on the other side you have bacterium with the plasmid, and you cut it with the restriction enzyme and you add that part to your plasmid, leading to the introduction of recombinant DNA into yeast cell. Then you have your recombinant yeast cell here, and from this recombinant yeast cell you produce into large amount in the fermentation tank, uh, and then your HP antigens will be start producing. Your hepatitis B antigen will be start producing in these bacteria. Then recombinant yeast will multiply and produce HP antigens in fermentation tank, and then you purify these parts, extract them, and then your HP vaccine is ready to go. Further, how commercial and pharmaceutical products uh, are possible in this from the health perspective. First is your insulin. So insulin controls glucose level in humans. So with the help of recombinant DNA, uh, cloning of human insulin and put it into the E. coli, you get your human insulin of by recombinant DNA technology and put into the E. coli. So you can have large amount of human insulin. So you avoid by getting products of animal slaughtering. So you don't have to cut animals to get insulin. You can do it in vitro with the help of recombinant DNA technologies. Okay, that's the same slide. And also you can produce human growth hormones that we have studied in our previous slides. Also that how you can add human growth amino acids into a sequence, and then you can grow put it into E. coli and grow the whole thing. So human growth hormone is a homing polypeptide having 121 amino acids and 2 to 150 Dalton molecular weight it is. Its role is in the growth, regeneration and differentiation. example, dwarfism treating by injecting these. So the people who are dwarf, we can increase their size by injecting growth hormones. So this is one example shown to you, right, how it's done as a method. Sequence for signal peptide, sequence for 160 amino acids for HGH. So the eCORI sequence for 24 amino acids, deletion of sequence of for 50 amino acids. So you delete those 50 amino acids, 26 and 24. Yeah. And then sequence of 167 amino acids of human growth hormones. You just get that one out of them. Then you encode with the 24 amino acids of human growth hormones. Complementary DNA, you ligate them together. Then you have a vector genome and you insert them into your E. coli for further growth. This is how you make your production of recombinant human growth hormone. So you can also grow interferon. Interferons are the group of proteins that interfere with the viral multiplications or replication by recombinant DNA capable of making interferons, alpha component of which have role in curing lymphoma and myelogenous leukemia. So these are all possible with the help of this. We can produce antibiotics. Artificially produced antibiotics are also available. They denature harmful living pathogens. For example, penicillin was found in 1928. Monoclonal antibodies are also possible with the help of hybridoma technology, in which an antibody of specific proteins are produced by the immune system in response to the presence of specific antigen. So monoclonal antibodies produced from a single clone of antigen. That's why it's monospecific in nature. So applications are to diagnose many diseases, pregnancy, and treatment of cancer. So here you have your antigen. You immunize your mouse cell and you isolate the immune cells out of them. Then this is your antibody forming cells. Then you fuse them your tumor cells and also your antibody forming cells. You fuse them together. So this way you have your hybridoma is made, and this hybridoma is screened for production of desired antibody. Tumor cells we choose because they produce in large amount. They don't stop. So afterwards, antibody producing hybridomas are cloned. Then this is your clonal expansion. Then further monoclonal antibodies will produce afterwards. Okay. Molecular diagnosis of diseases. Uh, Infectious diseases diagnosis mainly depends upon isolation and identification of pathogens, which may take several days. Okay. Uh, Development of diagnostic kits are also possible to identify pathogenic organisms by knowing the organism specific DNA sequence and has rapid specifically and correct diagnosis. So many diagnostic kits have been developed for AIDS, cancer, foot, mouth diseases, tuberculosis, and also recently for the COVID also. So COVID technique also work with the same technique with the positive or is it negative. So they see the results like this with the help of RT-PCR. You put your specimen there, drop off your specimen, complementary DNA, and then they tell you instantly whether it's a positive or negative. And it's not worth 4,500 rupees. One of my relatives did it, and it's very cheap. So it's all actually, I have to open it with you. It's all politics. That's people are playing with us. So they make this. They are doing these tests. They're making the money out of it, and making such expensive tests. They has to be. Their real amount should be not more than I would say 500 rupees. The real amount, because these tests are very cheap afterwards, but I don't know why they made so expensive. Anyhow, what is going on, we cannot stop that anymore. So further, based on ELISA PCR principles, for example, In HIV patient, diagnosis is conducted by antibody test using recombinant HIV proteins to measure antibodies in the body that proliferate when there is a HIV infection. DNA test uses reverse transcriptase polymerase RT-PCR to detect presence of HIV genetic material, and this technique was developed using recombinant DNA molecules analyzing the genome sequence. So this we also know DNA fingerprinting by Alec Jeffries, who have developed this technique. Every person has their own finger patterns that differ from other individuals. So there's a possibility to alter these patterns, but specific principle is unknown. So fingerprints are detected on the basis of highly polymorphic genes, that is VNTR. So it's used in the criminal identifications, child parentage establishment, and also for the detection of the racial groups. So that's we have done in quite detail. I will not go through with this technique. So we know about how DNA fingerprinting is done. Then somebody was asking me about gene therapy. Gene therapy is mainly done either you put transform cell into the body, or you add a transform vector itself. So you can use either way to put the improved gene into body to see the effect. So that's how the gene therapy works. So either you can inject functional genes into the cell to replace missing or defective genes in order to correct genetic disorders, or either what you can do is you can insert the cell usually that does not function instead of carrier called a vector genetically engineered to the deliver gene. And gene therapy may be done in vivo and in vitro, but it has some side effects, having some toxicity, inflammation, and even cancer could happen to you. So there is a direct delivery and cell-based delivery. In the direct delivery, therapeutic transgenes um, is packed into the delivery vehicle, such as virus, then injected into the patient directly to the organ, let's say liver in this case. On the other side, in the cell-based delivery, you have a therapeutic transgene, and then on the other side, you have a therapeutic transgene is packaged into delivery vehicles such as virus. And then the therapeutic transgene is introduced into delivery such as stem cells that is often derived from the patients. Then you mix them together, then you have a genetically modified stem cells that are multiplied in the laboratory. Then you re-administrate to the patients after having the stem cells with a therapeutic transgene into it. So there are two ways you can do this gene therapy. So what are the applications? So okay guys, to continue further, so there will be 5 minutes break now. So this will be too much for you. Suddenly there are so many things going on. Just have some relax, get something to drink, and then see you in 5 minutes. Okay. So welcome students. Um, so we're done with the health part. Now comes to the environment part. How recombinant a technology could be help in environment. Basically, it will let us know biosense that how much content pollutant is present in our environment and also to degrade that. So it can help in both ways to maintain the environment. So we can use recombinant DNA technology in an environment to clean up the environment or to measure the presence of hazardous compounds. So application of recombinant DNA technology in environment in this basically uh, technology in the environment you can remediate the environment pollutants by using some there are some harmful chemicals that are not digested and degraded, so they can be partially degraded such as TNT chemicals. So in this, nitrogen is reacted with oxygen and superoxide is formed, which is a toxic. So you knock out with the monodehydrogenase ascorbate reductase gene that will create resistance against these TNT genes. So with the help of monodehydroascorbate reductase gene, you can create resistance against these TNTs. Furthermore, arsenic particles are toxic and they are present in the soil, that is harmful for the plants. For so PA PVACR3 gene PVACR3 gene is inserted in Arabidopsis, so which is a model used being in plants, and it will give rise to tolerant plant which is tolerant to arsenic. So in Arabidopsis thaliana, reductase enzyme is present which reduces the arsenic, and phytochelatins resist the movement of arsenic in root cells and phloem cells. That's how you can have a arsenic resistance plant with the help of PVACR3 gene. Furthermore, to take it, waste product from agriculture such as cellulose, which is quite hard to decompose. So you can first what you can do is convert that cellulose into sugar for the breakdown of cellulose, and this is possible with a cellulase enzyme gene, and you put them into the vector, incorporate the bacteria, and you can have expression of these genes will enhance to degrade the waste that contains cellulose. So you can degrade it by enhancing into the vector of your cellulase enzyme gene, which we can degrade from your cellulose to sugar for smaller sugar breakdown. So by recombinant DNA technology, plastic degradations can be enhanced by genetically modified organisms that degrade oil spills or organic waste. Genetically modified strains of Pseudomonas putida are able to degrade these chemicals in oil spills. They could be used as a bioindicators. So bacteria have been genetically modified as a bioluminescence that gives off light in response to several chemical pollutants. So this is how they could be also in bioindicators and also degrader of plastic. So they are being used to measure presence of some hazardous chemicals in the environment. Also, genetic sensors in chemical contaminants is going under trials, and also include sensors that could track pollutants naturally degrading in the groundwater. So this is all possible with the help of these recombinant DNA technology. For example, how it can do. So mercury resistance gene MER or the toluene degradation gene TOL. So both are linked to the genes code for bioluminescence within the bacterial cells. And these biosensors can significantly low levels of inorganic mercury or toluene that are present in the contaminated waters and soils, emitting visible light measured with fiber optic or fluorometers. So we are done with the health part, environment part. Now comes the agriculture parts. So recombinant DNA technology can be used for insertion of genes in plants, not only from related plant species but also from unrelated species such as uh, microorganisms. So it could help to create GMOs, genetically modified plants, GMPs, like transgenic plants, such as higher yield with nutritional values, increased resistance to stress and pests. The application is that it helps to improve yield, development of stress tolerant plant. Transgenic plants could be also a source for biopharmaceuticals. So recently, one of my friends in Bangalore, they have opened a company to create a COVID vaccine from plants itself. So they use Arabidopsis thaliana as a model plant, and they have injected vaccine into them, and they want to grow instead of growing in bacteria or in animal cells, they want to grow in the plants. I forgot the company's name, but they are looking for grants actually at the moment and to make it a big idea afterwards. If they're successful, then they could create with the help of transgenic plants huge amount of COVID vaccines in upcoming future. Because that will be an ideal. Why? Because you need little bit space of growing your plants in small chambers, and you can grow them on the trays within the same place, and you can have huge amount of vaccine afterwards within the small time. So that's why it is that's the future. So you can plant with the improved yield. So was once you can insert genes to increase the yield of your plants. Genes are inserted. So researchers at the Japan National Institute of Agrobiological Resources, they added maize photosynthesis genes to rice. So it has increased yield by 30%. Yeah, the yield was increased by 30%. And to continue, maximum Mexican scientists have recently genetically modified plants to secrete citric acid from their roots. Citric acid could be also secreted from the roots of the plants. And in response to slightly increase in acidity, minerals bound to soil particles are released and made available to the plant. Stress tolerant plants. So plant resistance to environmental stress. Recombinant DNA technology is used to develop crops that can tolerate abiotic stress. So there is a possible way you can tolerate the abiotic stress. So genetically modified tomato and canola plants can tolerate salt levels of 300% greater than the normal one. So here you can see in the within the salinity in the tolerant plant, they are growing very well. The one which is susceptible, they got really rusted and a lot of smut is there, are not able to grow much. So that's the best part of growing your stress tolerant plant with the help of rDNA technology. You can grow herbicide resistant plants. So Roundup is a herbicide. It kills almost all species of plants. So using recombinant DNA technology, we can create a EPSP synthase gene into the crop plant. So we can grow cotton and soybean with the help of this. Furthermore, insect resistance plants could be grown, like cry gene, also known as BT genes, from bacterium Bacillus thuringiensis are isolated, and then plant is modified using this gene. So these genes will be resistance against the insects. So mainly used in the cotton, rice, maize, potato, brinjal, cauliflower, cabbage. In all these vegetables, it has been used quite widely with BT genes. You can also produce some disease resistance plants that are modified to produce resistance against disease. Example, tobacco was first modified to produce resistance against tobacco mosaic virus, TMV. So this is the example of the same, how it is done. Further production of biopharmaceuticals. So recombinant DNA technology used to produce a plants that will generate a seed and express a desired therapeutic plant. Then seed stock is used for producing the desired protein. The desired proteins can be extracted from the seed, and then in corn, biopharmaceuticals are produced with the help of this. You can also produce edible vaccines. These genes encoding antigenic proteins and can be isolated from the pathogens expressed in plants producing antigens and can be eaten just as edible vaccine. So in banana and tomato, we have edible vaccines are made already. Then our production of secondary metabolites. So Arabidopsis plants are modified to release polyhydroxybutyrate, which is a biodegradable plastic. So you're also producing a biodegradable plastic with the help of Arabidopsis. So I will stop here and like to see all going fine so far. No, bache, today is not the last class. Thank you, Sakib. Thank you for review. We worked. So one person could send me who is the you can say one person always send the review to the editor also. The one who did everything, who was a team leader, please send it as a way. But the synchrotron is the bigger part of producing X-ray crystallography of your X-rays. It's a synchrotron is the size of whole football field where it sends these electrons in a very X-ray, sorry, in a very fast rate. So it's better than the normal X-rays machines. But yeah, yeast is very useful aspect. When I was in a conference in USA, so most of them they were using yeast as a model to identify the various data because it's very fast. You can get that, and its genomic data is quite similar to human data. So whatever the data that you find in the yeast cells, you can say that okay, whatever you send, you can search for the same for in the humans also. Last date बच्चे project submission is uh, just 3 days or 5 days after the from tomorrow onwards, 5 days maybe maximum. So बच्चे if the project is ready from her side, she will take its her name is this is Damandeep Kaur, and she's a MSc in bioinformatics from Lund University in Sweden. So if everything is fine, she will take your class tomorrow. Otherwise, I will take your class. Oh, who can? So far I know for the Nobel Prize winner, mainly it's done in the Karlonsika Institute. In Karlonsika Institute, uh, they check the worldwide. It's in Solna. I was there also once. Karlonsika Institute. It's a medical university. There they choose all the. Uh, you can check it there, and you can also work there if you want. Just go to the career website, and you can look for PhD positions, masters. You can also do masters there. It's a bit expensive though, but if you take a loan, it's okay. Your future will be secure. So I will highly recommend to go for this university, Karlonsika Institute. Not everyone get the admission, but if you get, it's a lifetime windows open. Okay, I will continue now. In the case of industry, application of recombinant DNA technology in industry. So traditionally, industrial microbiology is merged with the molecular biology to yield improved recombinant process for industrial production. So primary, secondary metabolites, protein biopharmaceuticals, industrial enzymes, they're all formed of industrial usage. So with the help of primary metabolites like amino acids, nucleotides, vitamins, solvents, organic acids, they are all possible to grow with the help of recombinant DNA technology. So their production is related to energy production and substrate utilization, which are essential for the growth. So vitamin B12 is one of the protein that is being produced commercially for by direct fermentation utilizing the fungus named as Asbia gossypia. Vitamin B12 is produced by direct fermentation utilizing Streptomyces species also, such as Streptomyces griseus. Vitamin C could be produced utilizing gluconobacter oxidants, and P carotene is produced by members of Coniferaceae family of phycomycetes. So with the E. coli and cloning vector PBR322, the famous one, they were used to increase the genes for production of amino acids like glutamic acids, lysine, phenylalanine, valine, and also amino acids like L-glutamate, L-lysine, mostly used as a feed for additives. So here is showing you the golden rice technology, how it was produced. Um, so you have daffodilus plants, and these are, and you have bacterias, Erwinia bacteria. So the genes that give golden rice its ability to make beta carotene in its endosperm comes from the daffodilus and bacteria called uh, Erwinia uridivora. So they are mixed together. You got the genes from each of them, and you have different plasmids. You can see from the plant, you got green and red, and from the bacteria you got the yellow. And you put them into the agrobacterium. So these genes, along with the promoters, are inserted into the plasmids. Then uh, occur inside a species of bacterium known as Agrobacterium tumefaciens. So this is Agrobacterium tumefaciens is highly used in the genetic engineering. This is the most, like in the case of human study, if you want to study, then mostly yeast cells are being used. Then in the case of plants, Agrobacterium tumefaciens are used as a bacteria. So these agrobacteria are then added to a petri dish, a dish containing rice embryos. With the help of these rice embryos and these bacteria, they will combine together and they affect the embryos, and they also transfer the genes that encode the trans instruction for making beta carotene. So they will be code now to make how to make beta carotene. So they will be coded for the same. Then these transgenic rice plant must now be crossed with strains of rice that are grown locally and suited to particular regions, climate, and growing conditions. So you can breed with the other local plants, and then your variety will be start growing, producing provitamin A, producing rice embryos. So you will have a vitamin A producing rice, highly growing in the local area of the climatic conditions. So that's how the first golden rice came to the play role. So enzymes are biological molecules that catalyze and increase the rates of chemical reactions. So recombinant enzymes plays vital role in the food industry, such as the amylases from fungi and plants make highly fructose corn syrup, and from proteases you can make in biscuits manufacturers. They reduce the protein levels of the flour with the help of proteases. And this is to tell you about how the amylase production is done. So you have your double-stranded plasmid DNA expression vector promoter sequence, cut DNA with restriction nucleases, then insert amylase coding DNA sequences. So your amylase is being added there, then introduced into the recombinant DNA, and then from there, with the help of mRNA, then overexpressed amylase is produced. So that's how your overproduction of amylase is done in with the help of uh, lower the yeah sorry. So that's you can increase the overexpressed amylase. Sorry. I just got lost. Then you can in the cheese factory. So chymosins are derived from rennet used to manufacture the cheese. So amino acids and peptides that can be used for production used by yeast for fermentations. Proteases split the polysaccharides and proteins in the malt. So first what will happen, prochromosines genes are isolated from the young calves. Then they are transferred to the plasmid, and this plasmid is then introduced into the microorganisms. Then on expression, prochymosin is activated into chymosin, producing 100% pure chymosin. So your from prochymosin, your activated chymosin is done producing 100% pure chymosin. So this is how cheese production is done in the recombinant chymosin. So it might be small for you. So first gene for chymosin is isolated from the cow DNA, and the plasmid DNA is cut open with restriction enzymes, and this chymosin is combined with the plasmid with the help of lipase enzyme, and then the transformation is happening in the bacterial chromosome, where the protein is expressed in the bacterial cells. So chymosin is being produced in the bacterial cells. Then you isolate this chymosin protein, and then isolate chymosin is used to make cheese. Then further, so here your recombinant DNA technology is also highly being produced in this case also. So all the small startups in Bangalore, in northern India, or anywhere in the world, they are mainly done with the help of these small recombinant DNA technology. You have to just overexpress one thing. You need to set up lab. You need a little bit of funding, and then voila, your first production of enzymes are already started. So you don't need because you are all now biotechnologists. You know all these techniques by your heart. It's what you all need is to see their practical application, and there's a highly demand in the market, because don't think that you have to go to each store for selling them. It will be absorbed automatically later on. So if somebody is interested in startups, to make your own company, to be an entrepreneur, to be master of your own field, go for it. Go for recombinant DNA technology and choose whatever the field is suits you best, and start working on it. And that's how the Biocon started, with the Pepase enzyme, Karun Majumdar Shah. So she started it in her garage one day, and now yeah, she's queen of biotechnology. I have to agree. She's a queen. Then biofuel industry, cellulases used to break down cellulose into sugars that can be fermented. In molecular biology, restriction enzymes, DNA ligase, polymerases used to manipulate DNA in genetic engineering is important in pharmacology, agriculture, medicines, could be used in the forensic science also. So thank you very much. So we are done for today, I guess. No, we have one more lecture. So I will just uh, check if everything is fine. But we are done for today actually. Uh, so now I will take you to the next part. Do you want 5 minutes break? Just take 5 minutes break. Then we can start with good scientific practices, GSP. Because we are done with all techniques, everything is done. But when you comes to work in reality, you should understand that you have to work a clear, neat, without plagiarism, without any sabotage, and produce data genuinely good and something original. And no fabrication is done with your data, with the Western blot data or mRNA data. So you have to take care of that. So we will deal with that topic now, with the good scientific practices. Just take 5 minutes break. I need to get some water bottle. Then we start at 9:23 or so with a fresh start. So, students, I'm back to start with the next topic, good scientific practices. Mm, Most of the time, when you're starting your PhD or your master thesis or anything, we were not told about how a scientific world works, how the academia works. So you have to be very careful when you're going to start with your new research, how things are working, because sometimes groups are not so good as they should be. So carefully ask around when you're going to start your work any somewhere, how things are actually. How is the past record of your PI? How is the past record of your team members? How many publications they have? Does they have any bad thing attached to them? Check all these things, then start your group. Start with your things. So to start with, so there are also many misconducts has been done in recent years, um, related to in cancer field or into the rainforest, or by Japanese, some they did some false things. So there are a lot of misconducts has been done in research fields. So some FAQs for the good scientific practices. So does my PhD projects or your master project you will do require ethics approval? Do I need to store my original data to the scientist? Am I allowed to eliminate outliers from my data? There are some outliers also. Is using sentence from another paper already a plagiarism? Under which conditions can I use figure from another paper? Who qualifies for co-authorship on my paper? Whom should I contact if I suspect scientific misconducts? So these are the various questions that might be arising when you're going to start your research anywhere in the world. So, bache, this topic might be it's good for your future, but I want to touch this topic because no one wants to touch this topic because this is so sensitive topic is. So that's why I'm bringing it to you. So we will deal with responsible conduct of research, such as values, legal frameworks, data management, how you have to keep your lab books, some case studies we will do, and how misconduct is done by fabrication, falsification, and plagiarism. So shared values for the responsible conduct for research are honesty, accuracy, efficiency, openness. So these 4 points have to be take care. So in honesty, conveying information truthfully and honoring the commitments. Accuracy, reporting findings precisely, taking care to avoid errors. Efficiency has to take care of using resource wisely. And openness, let the facts that you are telling about should speak for themselves. Avoid improper bias. So, section one, in general, principles, rule for good scientific practice is to respect the rules of good scientific practice. Yeah. Preventive measures, scientific misconduct should not be there. Contact person in the case of suspected scientific misconduct. If any scientific misconduct is done, then check with the persons who could help you in that case. Procedure in the case of suspected scientific misconducts, check the committee within your university who are regulating the scientific practices, who are formulating the investigation committee. And additional marriage, you can have storage of records, what you have done. Then, yeah, check the rules of authorship, how things are working out. Further protection of human subjects, local ethics committee, respect for person should be voluntary. When somebody is working with the human, this clinical trials are happening. Whether the clinical trials are happening voluntary or not, just check by that when you're working research in that part. Or so is there any vulnerable subjects are happening instead of we are using chimpanzees for experiments, which is not allowed, then it's also not good. um, Justice should be given to the benefits and risk equally without prejudice to the particular individuals or groups. So welfare of the laboratory animals, if you can replace your animal models with the cell culture techniques, then go for it. Reduction of methods using animal models, that will be even better. refinement, elimination of reduction of unnecessary pain and distress. Because when I was working in my PhD time, so I was into the monkey research institute. So there they were giving injections to the baby monkey on every third or fourth day. So these monkeys were living with the mother. So whenever the scientists coming to give them injections, mother got really frightened and they keep their kids to themselves. If they're coming up, so they were like making a lot of noise and making things uncomfortable for everyone. So I've seen the pain, how when you are taking these animal models, how their mother, their family as a together, they behaved. So just be careful. So that's why our cell culture techniques are quite workable. Everyone wants to work on cell culture. That's why, because it's very painful to work with animals and it's not humane to do so. Of course, we are doing it for the human beings, for us, but in the price of others' life. So you have to see, you have to count for yourself which is best in that case. Then rules for good practice for doctor supervisions. Uh, so do not fight. First of all, like in this picture, they are doing so. Uh, so doctors shouldn't, students should inform the supervisor immediately if any problem. Expert supervisors should be confident in primary contact person of the doctor student. Thesis committee members should be there for you to check the things done, that everything is done in right manner. In the data management, university funding, data source will be taken care for. Data protection has to be there. Data collection has to be done nicely. Data retention and sharing then later on. Like I show you my old data, that how I produced my PhD. Then it's been published in the university library, so everyone can use it. Everyone can read it, and it's available for retention, protection, and sharing. Uh, and then rules for good lab notebook. That's very important, because lab book is kind of a bible to you. If you are maintaining a good lab book, wherever you're stuck in experiments, your results are not coming up, just go to your lab book. You can see in the past what have you done, and check your things done how it is. So this is a lab book by Darwin, in the phylogenetic studies that he was doing. He's checking that about that. So notebook should be bound and paper durable. Pages should be numbered. Notebook must contain remain on site, should never be taken home for any purpose. So all notes should be easily legible and in permanent blue or black ink. Never erase or white out a data from your notebook. Pages must not be erased from the lab notebook. They should be remain there. It should have a content table of contents. Entries for each individual experiment should be there. Data, date of entry, descriptive title, purpose, methods, results, conclusion. So maintain your lab book like this. So whenever you got a chance to work in future, I think you are mostly BTech students or MSc students. So wherever you got a chance, just try to maintain your lab book like this that I'm showing you. So this will be really good for yourself actually, for your taking care of your own well well care. So this is one example. So this is shown you as a table of contents with the session 1 vacuum system, electron beam description, beam in magnetic field. Then session 2, page number is there. Then on the side there's a title, there's a date, there's the name of the lab partner, then plain task by task. So each task has been shown in very nicely and in a good manner. This is something with some law they're talking about. So some case studies of falsifying scientists that they were dealing with. So you can see that failing to present data that contradicts one own open previous search. So they were like around 6.5 percentage to people were having this kind of things. So also changing the design methodology or results of study in response to pressure from the funding source. So they were like 20 in mid career. People were like 20% people were doing this. So these are the various misbehaviors you can see in previous 3 years that people are behaving like this. So, research misconducts could be fabrication, making up your data, falsification, manipulating, changing, or omitting data, plagiarism, steal another's personal ideas, results, words without giving them appreciation, sabotaging your data, like adversely affecting the research activities of others, violating the recognized rule of authorship. So, these are all possible in this. Fabrication is making up, inventing data or results, or recording, reporting them. So falsification is manipulating, changing, or omitting data. So here you can see uh, Minister Triton of Germany. He was actually walking through, but people made a advertisement that he's a part of the Reuters. He's influencing the Reuters to do so. So that's how you can do with the data also. So what's in picture is not it looks like. So it's the same way in the Western blotting with the protein analysis. So you can you can play around with this gamma ray. So you can bring it this down a bit here. You can remove the background and you can enhance your protein expression that I was talking about on with the CTGF data. So which could falsify the things that you are in that data. So last, so these are the gross uh, misrepresentations done. So originally the person has this data. Yeah, originally the person has this data. Uh, just wait a second. Uh, so there are three rows: one, two, three, four. So you can add or delete bands. So band 3 uh, deleted in lane 3. So band 3 is deleted. Okay. So somebody published this in JCB, Journal of Cellular Biology. And then band 3 is added. So initially there was no band, but there was a band here, but the person needs a band here also. So what he did, he first moved the upper band, then he added this band. So this is not good. This is not good if you are playing around this way of a game. And this is a gross misrepresentation. This is your loading control A, loading control B experiments are being done. So duplicated panels. So experiment A has this data, experiment B of the same has this data, but loading controls are different. So they are also doing in this way. So also adjusting the intensity of a single band. So this single band has been in original it looked like this. In the manipulated image it looks like this. You see? So you are playing around with the data. So this is the contrast management. So this is the original image. You can with the removing the contrast you increase expression of your protein like this, which is also not good. But originally the protein was of this much size, that's it. So you increase the expression of a protein so much high just by increasing the contrast, adjusting the contrast. So this is original image and this is the manipulative image. So this was highly expressed, but now both looks quite okay, quite normal. So this is done by Photoshop. So don't do these things in your case. Photoshop, deleting, contrast, all these things. So original image looks like this, and this is the manipulative image. So they added these dots extra on top to enhance it. So this is the cellular data and this is manipulative data. So these and also take care of plagiarism that whatever you're gonna produce, you should not do it on your own. You should not take somebody's work and then start doing your own research with that. So take care of that. So the nucleus, so let's say the DNA in an eukaryotic cells is sequestered in the nucleus, which occupies about 10% of the total volume. So what you can say is the DNA in eukaryotic cell is sequestered in the nucleus, which occupies about 10% of the total cell volume. So you wrote the same way. So this is plagiarism. You didn't change anything. So so DNA in this case, which occupies about 10 of total cell volume. So but when you write this is okay that uh, you have put you have given the author's name, but you choose the same line. But then uh, in a eukaryotic cell, the nucleus contains DNA and make up approx 10% of the cell volume. So this is paraphrasing. This also come under plagiarism. So you have to write like this. DNA in eukaryotic cell is sequestered in the nucleus, which occupies about 10% of cell volume. Then Albers et al. 1994. This is direct quotation. So correct quotations would be in an eukaryotic cell, the nucleus contains DNA and makes approx 10% of the cell volume. That's it. That's the correct conditions to do so. So this is how I told you about how which sentence has to be copied, which is not, and how you have to write the terms in this way. So that's it. Just go through them. These are the sources. So that's it for today from my side. So, writing a PhD thesis is also a lifetime opportunity that somebody has it. You learn a lot from it. Actually, you learn a lot of how to manage your time, your patience, bringing new ideas. All these things matters a lot then. So, I would say overall, it's a good thing to write your PhD thesis. Just go through some read from your previous lab friends that who have wrote the thesis, how they have wrote. Just get influence a little bit. Okay, this is the parameters I have to take, but you have to write your own language, in your own things with your new experiments. So no one has done that in the world before. So it's like writing a book. Sorry, writing a book which no one has wrote before, but you're writing it for the first time. So yes, there will be bioinformatics class tomorrow for sure. I will take care of that. So if there is a group, send one person can send me for the whole group with the name of them. So I will take care of that. But just software toolkit. So there was software toolkit. Basically what I have shown you with the things here with the videos and things. I think that was the main thing that was required in this case. Main idea was to convey you all ideas how things are working out. But if somebody is teaching you bioinformatics, there might be you need some softwares. But in our case with MBBT, there was not much needed. But I send you some protocols that is most required was there, some videos, some presentations. So all these things were quite necessary to make this workshop workable. So yeah. But as I said, summary of whole project will be nice to keep it with you in the future, because once you finish this workshop, you will have the certificate. It will be in your CV. Okay, that's good. But you will forget all these things. You will forget after even 1 week. I'm damn sure. You will forget each of these things here. So it will be nice to keep a record of all things that you have done in the form of assignments that I have given you. So just do it. Just do it. It will not take your 1 day. It will not take even 1 day for you. It is six hours job. It is 6 hours job. If you sit once, go through the videos, and just tap what you have understood. Go through the presentations, open the presentations, and that's it. So just do it. Just it's for you. And review, I have seen it's very highly good reviews I'm having. So I really liking them. So internship projects is there. So send me each thing on the email so I will save them for future. So I will go through over the weekends and let you know how things are. Okay? Any further questions? This is a doubt session is going on. So, bache, thing is, I will personally see according to your work which site will be best to publish for your review articles, because you also need your affiliation to your university to publish that, and also need affiliation from your boss somewhere. Either one of your boss could help you. Oh, sorry, boss. I'm saying professors in your university. You can collaborate with them who's working in the same field, or or you do it on your own. Then last date is 3 to 5 days from tomorrow. So today is 3rd of September. So 1, 2, 3, 4, 5, 8th 8th of September or 9th of September. Send me in between them, 8th and 9th. 8th to 9th September. I just share you on the Viber everything now itself so that we are not late later. Day 10. Production. There you go. So, so yes, yes, it will be. It will be. You're welcome. You're welcome. Let's see if I will be there tomorrow or ma'am will be there. Someone will take for sure. If I am not there, then I have to say goodbye to all of you. It was very nice. Otherwise, I will say the same things tomorrow also. If ma'am is not there, so it's dicey. You're welcome, bache. You're welcome. Welcome, everyone. Welcome. I will be happy if, with the help of this workshop, you stand to your dream come job. Actually, it's good you did. It should not be just one certificate that you are here for. This is a workshop. If you can land to your prescribed things that you were looking in the future, either your masters, your PhD, or your bachelor's, whatever things that you are, you I want you to take all the things that you have learned from these workshops and apply them for your future. You can say strongly that I'm an MBBT expert. You can ask me anything about MBBT, because I try my best to teach you things in a simpler way. So with a lot of videos and lot of virtual labs, uh, yeah. If you're still there, I mean, our flow cytometry virtual lab is left. Would you like to continue that? I found it just today. There's a flow cytometry workshop. Okay, I can show you. We still have time. We have 15 minutes left. We still have 15 minutes left. So I can show you one review maybe. Pancreatic organogenesis by Tushar. So first of all, he sent me in the PDF format. That's very good. And he write it in a thesis way. Somehow the okay, he made everything on the left. fonts are not in the centered, so these things have to be taken care. So he put this declaration. That's nice. So he put acknowledgement. Then abstract is there. Then comes the introduction. Then the main part. Then yeah, and each part with the detailed references. Very nice. Some methods, replication, isolates. So something problem with the paragraph lining. Say is okay. This is your new start. Conclusions. So with the, at the end references. So this is very nice. Okay. I have to go through this. This is a lot of work there. I can see. That's good. I like it. I love it. So I feel like my training given to you has been really worked out here. What I ask, I'm getting from you students. There's also one more I want to show you. CRISPR-Cas9 system, the Pandora box for modern biological review. So authors, uh, okay, two authors are there from BTech students. Then nicely, everything is in order, very nicely done with the abstract, with the introduction, then all the methods, then figures are there. Okay, good, very nice. Uh, with each references being quoted very nicely with these. Very good, bache. Wow. So at the end references, then conclusion and future prospects. It's indeed very nice. I really like it. It's good to the eyes actually, that somebody did some efforts and it's working out. So that's really good, really good. So um, if you're still with me, let's start with this flow cytometry workshop.
Flow cytometry is a tool that can be used to get information about your blood. In this virtual lab, you will use flow cytometry to do a complete blood count, diagnose a type of leukemia, a blood cancer, and look for any signs of cancer that remain after treatment. Blood is made up of liquid called plasma and multiple types of blood cells, each of which carries out a specific job. Select a blood cell to learn more. White blood cells help us to fight infections. There are multiple types of white blood cells, each with a different specialty. Red blood cells carry oxygen from our lungs to all of the body's tissues, and they carry carbon dioxide away. When we have a cut, platelets help stop the bleeding by allowing blood to form a clot. In a healthy person, each type of blood cell is present in predictable proportions. If the proportions fall outside of what is considered normal, it can be a sign that something is wrong. A complete blood count, or CBC, is one of the most commonly ordered lab tests. The test measures the proportions of different CBC tests used to be done manually by looking at blood through a microscope. In a few minutes, a technician can count about 100 to 200 cells. Let's see how many you can count in a minute.
So I have 1 minute.
An automated blood analyzer, on the other hand, can count many thousands of cells in just a few seconds. Its speed and accuracy make it a much more popular choice. The machine draws in a small amount of blood. Red blood cells and platelets have distinctive characteristics, so they can be counted with a method based on electricity. The differences between white blood cells are more subtle. To tell them apart, the analyzer isolates them and then uses flow cytometry to count them. The cells pass through a narrow tube in single file. One at a time, they pass by a laser. When the light hits the cell, it scatters. Across from the laser, a detector senses the amount of forward light scatter. Larger cells cause more forward scatter, and smaller cells cause less. Another detector senses the amount of side light scatter. Cells with a lot of internal complexity cause more side scatter, and simpler cells cause less. Computer software counts the cells and plots their light scattering properties on a graph. Since each cell type has a different scatter profile, we can use the graph to identify the cell types present in the blood sample and their proportions. Does this person's blood count look normal? Compare these results with those from a healthy person and note the differences. The plot doesn't look quite right. This person has many more immature white blood cells in their blood than normal. To get a better sense of what's going on, take a look at the blood. The results suggest acute leukemia, a type of blood cancer. The patient's doctor will follow up to see if these results line up with any symptoms the patient may be experiencing.
Okay, it's got stopped. I think it's got stopped somehow for same strange reason, but I will send you the link. Just go through them by yourself. That's the main thing that I want to show you. The rest we have done in quite detail this about this workshop. So just go through this link also. It's not working for strange reason. Okay, it's enough. That's I want to show you all. Okay, did I face any kind of racism? No, bache. In my 7 years, I never faced any racism. I was wearing my turban all the time with my beards, and I never faced anything like that. So people are very nice. Germans are very nice. So it's just made a stereotypes from the past, being Nazis and all these things, but they are not. That was their past, but it's not working anymore like that there. They are super duper nice people and helping you in all your difficulties. I still have my friends from there. So they even made I want to show you, it's personal thing though, but they made birthday with me online with the help of iPad. He made a cake and cut everything. So they are my friends from there. So they made this happy birthday cake for me, and they are one of the nicest people so far to my in contact with no racism thing in them. Nothing like that. It's all about quality, that if you provide them good quality, if you do everything positively things, then there will be nothing like that. So you have to just adapt to the new place, bring them to your culture, and just see, voila, it's a new life. Yeah. So, okay, if you have any other further questions, I would like to end the stream, and take care then, students. It was such a nice time. And yes, please subscribe to the channel, like, share with your friends, and go for it. Make this YouTube channel viral. Yeah. Software toolkit. Uh, Bache, software toolkit is something that includes all your videos, your presentations, your live, your virtual labs. So this is all I could include in the MBBT. So it was more, software toolkits might be required. Somebody is dealing within the Python language in IT sector, but in our case, we were not need of it. So yeah. So it was my personal reason, my family reason. I have to come back to India, and so I'm, yeah, something happened family, so I have to come back and start life from here itself. But you never know where life goes take us to. So be wise, be happy where you are, and get the most out of it. That I would say. Wherever you are, get the most out of it, and yeah. Okay, on that note, thank you very much, everyone. It was a pleasure seeing you all, and hope to see you in future workshops also. Take care, sleep tight, good night, and yeah, thank you very much. Thank you. Good night, everyone. Take care. Sleep good. Bye bye.